Hey guys, this is Malinki. Welcome back to my channel, Voice of Malinki. Today we will talk about HMPV. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. So nowadays, people are worried about this new virus. Let's talk about this. So first of all, the full form of HMPV is Human Metapneumovirus. Pneumo. That means from the name itself you can understand this virus has a direct effect on our lungs. It belongs to the family Pneumoviridae. It causes symptoms similar to cold. It is a negative sense single-stranded RNA virus. So what is negative sense single-stranded RNA virus? First of all, it is a RNA virus. That means it has only RNA and not DNA. And the RNA is single-stranded. Okay, so what is negative sense RNA virus? Basically, viral RNAs are of two types, positive sense and negative sense. Positive sense RNA can make proteins directly. That means it is just like normal mRNA. But negative sense RNA cannot make proteins directly. It should be converted into positive sense RNA first, then only that will be translated to make proteins. And these positive and negative sense RNAs are just complementary to each other. Okay, now let's talk about its genomic structure. The viral RNA encodes 9 proteins such as N, P, M, F, M21, M22, SH, G, L. The direction is from 3 to 5 prime. So this N is for nucleoprotein, P is for phosphoprotein, M is for matrix protein, F is for fusion protein, M21 and M22 these are for matrix proteins, SH is for small hydrophobic protein, G is for glycoprotein and A is for polymerase. And this is the main structure of HMPV. Okay, so it has the lipid bilayer and here are matrix proteins just below the lipid bilayer. Here we have SH proteins in this way, they are embedded in the lipid bilayer. And here are fusion proteins, this green one, fusion proteins F. This is the glycoprotein G, this one. And here is the RNA, this is the RNA. And in the RNA, you can see nucleoproteins, these blue color, these are nucleoproteins N. Here is the polymerase L and one more is there, this is phosphoprotein P, this is phosphoprotein P. So basically the nucleoprotein is a protein that is linked to a nucleic acid. Here it is RNA. And you can see from this picture that nucleoprotein, this N, phosphoprotein, this P and polymerase L, these are attached to the RNA genome. Okay, so this is the basic structure. Now let's talk about its life cycle and reproduction. So the virus gets attached to the epithelial cells of the respiratory tract using the G protein. So basically the epithelial cells of our respiratory tract are the main target of the virus. And virus gets attached to them via the G protein. Okay, so you can see this image. Let's say this is the epithelial cell. Here is the plasma membrane of the cell. Here is the virus. So the virus gets attached to this plasma membrane with G proteins. And G protein contains a hydrophobic region that helps to facilitate its binding to the host cell plasma membrane. Why? Since plasma membrane is also hydrophobic, since it has the lipid bilayer. So this G protein helps in this binding. Next step, fusion of the viral and host membranes is mediated by F protein. So these are basically fusion proteins, F proteins, and through those F proteins, the virus gets fused to this plasma membrane. Next step, RNA genome is released into the cytoplasm. So here is the RNA genome it gets released in the cytoplasm just like this. And we know that the RNA genome is already associated with these three proteins: nucleoproteins, phosphoproteins, and polymerase, right? Okay, that I have already mentioned. And this RNA genome acts as a template for positive sense RNA. So this is negative sense RNA and it acts as a template for positive sense RNA. So I have already said that negative sense RNA is incapable of making proteins. Hence, it should be converted into positive sense RNA. And these two are complementary to each other. So how does it occur? So the proteins N, P and L, they dissociate from the viral RNA and bind to each other to form the polymerase complex. So these three proteins N, P and L, these three proteins will be dissociated from this negative sense RNA. They will be in the complex and they will form polymerase complex. And that polymerase basically helps in the replication of positive sense RNA. And positive sense RNA is capable of translating. So it will make proteins, different proteins of the virus, and few of the positive sense RNA, few of them also make negative sense RNA as genetic material. So basically, from negative sense RNA, positive sense RNA will be formed, and from positive sense RNA, again negative sense RNAs will be formed. This is important since the virus actually has the negative sense RNA, not the positive sense RNA. So negative sense RNA should be formed as well as proteins should be formed to assemble the virus. And these proteins and negative sense RNA will be assembled in this way. And the matrix protein helps in this assembly and budding. After the budding, the virus will be released from the cytoplasm and will attack neighbor cells. So in this way, the cycle will continue. Okay, so let's talk about the detection. So basically, reverse transcriptase polymerase chain reaction or RT-PCR is used to amplify directly from RNA extracted from respiratory specimens. So basically, from respiratory tract, specimens would be collected and RT-PCR will be performed directly using RNAs. So in reverse transcription, cDNA would be generated from RNA, and if the person is affected, cDNA will be obtained, and if the person is not affected, there will be no cDNA, right? And bronchoscopy and chest X-ray would be performed to check the condition of the patient's lung. Okay, next let's talk about its transmission. So basically, the transmission of this HMPV is seasonal, That means it is more likely to occur during winter or early spring. It gets transmitted through respiratory droplets from an infected person's cough or sneeze or by touching contaminated surfaces, just like COVID-19 virus, coronavirus. And infants and toddlers specifically who are under age 5 and adults over 65, these aged persons are highly susceptible who have weakened immunity. So if you have a good immunity, it's not a problem for you. Symptoms: cough, nasal congestion, runny nose, fever, sore throat, wheezing, breathing problem, fatigue, bronchitis, pneumonia. Treatment: so no specific antiviral medication or vaccine is available till now. And rest, adequate hydration, nasal spray, inhaler, medications to manage fever, these are only used to treat a person. So this much of information I have till today, 8th January 2025. Hope you liked the video. Thank you for watching my video.